Hello everyone. In this video, I am going to discuss about numerical questions for ICAR JRF Plant Science exam, and this will be the part 2. So we are giving our time and effort to split these questions or split these videos into different parts because of the fact that these videos should be a kind of refreshment. So whenever you feel bored of reading, you should come to our channel and watch these videos, and again this video should be productive and helpful for you. So if you just like this content, please like, comment and share our video so that it will be encouraging us and motivating us to deliver more such content. Okay, so let's get started. So let us start with the first question, and this might be little unfamiliar to you all. So the question says a random mating population has an initial frequency of 0.8 and of A allele, let us take it as a A allele. So this is not small a, this is capital A, and a small A allele of 0.2. So what will be the approximate frequencies of genotypes capital A small a and small a when the inbreeding coefficient of the population becomes 0.30. So this has a direct formula. So it is talking about the F. So let me clarify it again, what is F and what is small d. So we have F, small d and also inbreeding depression, ID. So this is called as linkage disequilibrium. So this is the different concept, and now we are talking about coefficient of inbreeding. So students usually confuse with coefficient of inbreeding and inbreeding depression. So these two are the different terms, where the coefficient of inbreeding says about the probability of getting the alleles which are identical by descent or which are inherited from a same ancestor or identical by descent. So this is what coefficient of inbreeding means. And the inbreeding, we already know that this is because of the deleterious effect of accumulation of deleterious alleles during inbreeding. So performance will be reducing from generation to generation if the population is undergoing inbreeding. So this is what inbreeding depression means. So I will talk about the linkage disequilibrium in the later slides. Okay, so this was about the F, D and ID. Now they are asking about the F, it is 0.30. So you have to apply this direct formula, where for capital A and capital A, this is just P square plus PQF, and for capital A small a, it is 2PQ minus 2PQF, and for small a, it is Q square plus PQ into F. So if you just substitute the things like P square will be A into A, that would be like 0.8 into 0.8, so it will be 0.64. So PQF value will be somewhere around 0.048. So if you just add these two, so you will get 0.688, which is approximately equal to 0.69. Next, for second heterozygous one, 2PQ, 2 into 0.8 into 0.2. So this would be around 0.32, and minus the value of 2PQF is 0.096, and which will give us 0.22. And lastly, it is per PQF, this is again same, it is 0.048 plus Q square is, which is equal to 0.04. So this will be equal to 0.88, that is it will be 0.088. so we can approximate it to 0.09. so the correct answer will be third option. So you can remember this formula. So this is not a most frequently asked one, or it has been never asked in the exam before. So since the formula is easy, or formula is simple, and this is a simple concept, you can always remember this, so that if they are asking in the exam, you can solve this question. And the concept remains the same, that coefficient of due to the coefficient of inbreeding, so the inbreeding frequency is increasing and the heterozygotes frequency are decreasing. That's it. Okay. Second question. So this is about the blood group. The blood group problems are very important for you. So just give utmost concentration to this question. In case of Mn blood group, a sample of 100 individuals has 75% with M and 10 with Mn and 15 with N blood group. So which of the following will be the frequency of M and N alleles? So this is a case of codominance. So you can take it as like a frequency of allele M. We have a direct formula, frequency of M allele will be equal to number of m alleles in a population divided by total number of alleles. So what will be the number of total m alleles? So in the 75% with the m, that means they are having m and m, right? So in the diploid it is m and m. So each person is having two alleles, which is capital M and capital M. So it should be 2 into 75 to get total number of m alleles, plus even in the heterozygous there is one m. So there are 10 persons who are heterozygous for m, having m and n. So each person will be having one m allele, right? Other allele will be n, one allele will be m. So 10 will be having 10 m alleles. So you just plus it, like add with 10. So divided by total number of alleles. The population size is 100, which means like each individual will be having two alleles. So you should just multiply it with 2. So 100 into 2. So this will be somewhere around 150 plus 10. This is 160. So divided by 200. This is 160 by 200. So what will be the answer? It will be 0.80. So this was for capital M allele. So for n allele, this is again same. So it would be 15 into 2, since there are 15 persons with the n blood group, plus the heterozygous, which will be again 10 divided by 200. So 15 to the 30, 30 plus 10, 40, 40 by 200. It will be again 0.20. So this will be your answer. I hope like you understood the formula. Like for frequency, to find out the frequency of one allele, so you should just consider the number of alleles plus divided by total number of alleles that are present in the population. So number of specific alleles divided by total number of alleles in a population will give you the frequency of a specific allele. I hope like this is simple. So third one. So this is a conceptual question. Population was subjected to inbreeding for 5 generations, and how many generation of random mating is required for equilibrium to be attained for a single gene? So in this question, the single gene having two alleles. So this particular statement is very important, because so for the single gene and multiple alleles with single gene, what I mean to say is a single gene, whether it is having only two alleles or multiple alleles, when we are talking about single gene, in the both the cases it only one generation is required to attain the equilibrium. So you should remember this, like only one generation is required to attain the equilibrium, whatever may be the generation, even if it is 5, 10, it doesn't matter, whatever may be the generations, like number of generation doesn't matter if we are talking about a single gene. So it can be n number of generation. Even for the n number of generation, it has been undergoing inbreeding, even then it requires only one generation to attain the equilibrium. So the answer would be 1. So just make sure to remember this. This is the one of the important concept. So either it is for two alleles, they might also ask you for multiple alleles, or just two alleles. They have not mentioned anything here. That simply means that it is only made up of two alleles. So if they are mentioning about multiple alleles, you need not to get confused. Okay. So this is again the answer would be 1. Yeah. So we have the same question here. So in the population, the two genes are segregating independently, which are not at equilibrium. The value of D is 0.2, and what will be the value of D after 3 generations? So this is just related to the above one, but a slightly different concept. So here they are talking about linkage disequilibrium. So what do you mean by linkage disequilibrium? So the linkage disequilibrium is non-random association of the alleles. So this is what linkage disequilibrium means. So there won't be any random association, there would be non-random association of the alleles. So this is what it means. So according to the question, if the two genes are segregating independently, which are not at all in equilibrium, so if the value of D is 0.2, so what will be the value of D after 3 generation of random mating? So once after the inbreeding, if there is a continuous random mating, after generation, the value of D will be decreased by half, will be reduced by half in each generation. So if this is the case, if the value of D was 0.2 at first generation, at second generation it will be divided by 2, and again in third generation it will be divided by 2 by 2, and again in last third generation, after third generation of random mating, this will be divided by 2. So the answer would be 0.025. You just need to divide 0.2 3 times. So this is one time, second time and third time. So if you just divide this 0.2 by 3 times, you will get this answer, which is 0.025. So this would be your answer. So remember the concept, when they are talking about linkage disequilibrium, which is denoted by small d, this is a non-random association of the alleles. So when the random mating is occurring after inbreeding, so the value of d will decrease generation after generation. So how much it will reduce or decrease? It will be reduced by half. Okay, the value will be reducing by half. I hope this is clear. Next, so it's about hemophilia. This is also very important questions for your exam. So the hemophilia is a sex-linked trait, and in the population the frequency of hemophilic females was 0.16. So what will be the frequency of hemophilic males in the population? So if they are talking about the hemophilic females, they should be, since we know that disease is recessive, so the individual should have both the recessive alleles, so in the homozygous condition. So that would be small x, like x into x, like x bar into x bar, we can denote it. So the frequency is 0.6. So we, since it is let us take it as q square. So q square is 0.16. So what will be the value of q? It will be 0.04 or 0.4, right? It would be 0.4. So the q value is 0.4, which is the frequency of the x allele, right? So this is the frequency of x allele, which is equal to 0.4. So what they are asking, what will be the frequency of hemophilic males in the population? So we know that since it is in the hemizygous condition, we only require one allele for disease to occur in the males. So the frequency of in the males will be 0.4 only. Since only one allele is enough to get the disease in the males, the frequency will be 0.4. So but when we talk of females, it should be multiplied with another 0.4. Since there are two alleles, since the two recessive alleles are responsible for the cause of diseases in the females, we should consider 0.4 into 0.4. So otherwise, since it only one allele is enough in the males, it is 0.4. Next, so if the frequency of the hemophilic males was 0.1, so what will be the frequency of hemophilic females? So this is exactly opposite to the question that I have discussed before. So where they are giving that frequency of a single allele, single X allele is 0.1. So what will be the frequency of the hemophilic females? So they are asking about the complete genotype. So genotype is X bar into X bar. So what is the frequency of this genotype? So this is nothing but 0.1 into 0.1, which is equal to 0.01. So if they are asking for males, it will be the frequency of allele will be equal to frequency of X allele will be equal to genotype, frequency of genotype of this allele, right? So this will be equal in case of males, but when we talk of females, the frequency will be square of it. The frequency of allele or the frequency of a genotype will be the square of the q, that is 0.1 into 0.1. So this is how you can remember this concept. Next, a disomic number of organism is 14. How many chromosomes would be expected in the double trisomics? So again, reading this question, don't get confused with number of possible double trisomics. So this is, they are not asking about the number of possible double trisomic, they are just asking about number of chromosomes in double trisomic, like double trisomic conditions. We know that double trisomic can be denoted by 2n plus 1 plus 1. So we already know the value of 2n, it is 14. So 1 plus 1 will be 16. So there will be a total of 16 chromosomes here. So you should not be confusing this thing with number of possible double trisomics. Okay, so that would be n into n minus 1 by 2. So this is for double trisomics, and also for double monosomics it is same formula, that is n into n minus 1 by 2. So why both are same? Because in the both the cases either one is added or one is deleted. So that is why it will balance and both will have the same formula. The effect is same since it is getting added or deleted. So that is why the possibilities are same. So but when we talk of tetrasomics, it is just equal to n. Since it is 2n plus 2, the complete set is added, it should be either from the one chromosomes. What I mean to say is, for example, 2n is equal to 4, right? 2n is equal to 4. I'll just denote the chromosomes. So this is what 2n is equal to 4 means. If we are getting the tetrasomic conditions for these organisms where the chromosome number is 2n is equal to 4, either this whole pair should be duplicated or either this pair should be duplicated. So we have only two possibilities. So if 2n is equal to 4, what is the number of possibilities? It is always equal to n. Since only one copy, like one perfect set, can be duplicated, which is the condition of heterosomy, here there is only two possibilities. So n is, we know that the n is equal to 2 and the number of possibilities will be always equal to 2. So just remember, it will be always equal to its haploid number. Okay. So in the next question, they are asking about, in case of organisms having 5 pairs of chromosomes, which will be the frequency of gametes having 2 paternal centromeres? So let us decode this question first. So they are saying that in a cell there are 5 pairs of chromosomes. I'll just take one pair of chromosome now. So one will be obviously maternal and one will be paternal, right? So out of these 5 pairs of chromosomes, when the gametes are formed, we know that we will get 4 cells from a single meiosis event, or when the gametes are formed, and all the cells will be having, let us take it as 5, 5, 5, 5 and 5. So all the gametes will be having 5 chromosomes, each will be having 5 chromosomes, right? From the 2n it will be converted into n. So what they are asking is, in the gamete, what is the frequency that, or what is the probability that this 5 will contain at least 2 paternal chromosomes, whether it can be on the chromosome 1, chromosome 2, 3 or 4 or 5, but it should at least have 2 paternal chromosomes. So that is what they are asking. So if they are asking this type of question, I told you already, the simple formula is NCR p power n minus r q power r. So simple binomial expression or binomial equation will give you the answer, where n is equal to 5 here and r is equal to 2. Since we have to get 2 paternal centromeres in a single gamete, so r is equal to 2. So what will be the p and q? Since it will be equally divided, it will be equal to half. So if you just substitute the formula, you will get the answer 10 by 32. So I will just do it once and show you. So n is equal to 5, r is equal to 2. So 5c2 p power, p is 1 by 2, n minus r is 3 and 1 by 2 is 3. So this will be 1 by 8 and 1 by 4 into, so 5 into 4 divided by 2. So this is 20 by 10, like 10 into 1 by 8 into 4. So that will be equal to 10 by 32. So the 10 by 32 will be your answer. So at least for this reason, you have to first decode the question correctly, so that you can simply answer. There will be a very simple answer solution for these type of questions if it is related to binomial expression or binomial equation. Clear? So just remember the formula. If they are asking anything from probability or the frequency, most of the time it will be from the same formula, which is NCR p power n minus r q power r. Just try to remember the formulas, and you have to know like what is what, like n is what and r is what, and how to apply it. That's it. Next question. So in this, in an F2 segregating for 4 pair of alleles, the proportion of the individuals as the extreme as either parent which will be which of the following. So I have already explained this question in the part 1. You can go and just check it, this question, if you have missed it. So the answer would be 1 by 128. So if you just observe the options, you might be confused, because we will get the answer as 2 by 256. So seeing that you don't have this option, you need not to think that your solution is wrong. It is like just they are divided it, or they are just simplified it, right? Like it is 1 by 28. So you have to carefully check for other options also, if any other possibilities are there. So don't be in a tension after getting the solution if the solution is not matching with the answers. Just check that they might be given, they might have simplified the equation and given you. Okay. So the answer will be 1 by 128. Next one. Six babies are born in the hospital on the same day. What is the probability of that? Two of them will be boys and four of them will be girls. So again the same formula, NCR p power n minus r divided by into q power r. So n is 4, so sorry, n is 6 and r is, you can just take it, n is 2, r is 2 and p is always half, since it is boy or girl, q is also half. So if you just substitute, you will get the answer. You can do it on your own. I won't be saying the answer here, you can just do it on your own. And let me know in the comments what will be the answer to this question. Next one. So what will be the allelic frequency? The heterozygous genotype have small a will be twice frequent as the homozygous genotype in the random mating population at Hardy-Weinberg equilibrium. So here the genotype is not small a, it is capital A small a. Okay. So the what will be the allelic frequency if the heterozygous genotype capital A small a will be twice as frequent as homozygous genotype. That means capital A is equal to 2 into p square. So this is what it means. So capital A small a can be also written as 2pq is equal to 2 into p square. So this is the given equation. See, if you do not know any concept or if you are not able to apply any concept here, based on the option you can mark this mark correct answer for this question. How? You have to at least write this equation based on what they are giving. So they are just saying that heterozygous are twice the homozygous, so this is the equation. So this is 2pq is equal to p square, 2pq square, 2p square. Okay. So we do not know the value of p and q here. So how to get the answer? You just substitute the answers here. See, if you just consider p as a 0.6, so if it is 0.6 and this is 2 into 0.6 square, if p is equal to 0.6 then q will be equal to 0.4. So if you just substitute 0.4, so the LHS and RHS are not equal in the first case if you are taking the first option. But if you are taking the second one, just check the same thing. 2pq is equal to 2p square. So p is 0.5, obviously q will be also 0.5. 2 into p square is 0.5 into 0.5 and it is 2 here. So here the LHS is directly equal to RHS. So it simply logically it is again 0.5. Even sometimes if you miss the concept, you can solve the questions based on the options in this manner. Okay, just you can solve this logically, at least if you get the question and if you write the equation out of it. I hope this is clear. Next one. Which of the following is the number of recombinant joints produced by for each double strand break during homologous recombination? So this is the direct conceptual question, so which I have discussed in the class also. I hope like you might have also read this in the BDC, so you will be able to answer this question. So let me know the answer for this question in the comments. Okay, I will be seeing like how many of the comments I will get. So next question. So in this question, in an XXXY human, what will be the number of bar bodies? So they are asking about bar bodies. So this is also again very important. Just try to remember the concept. So the formula for number of bar bodies is equal to number of X chromosome minus number of autosomal sets in haploid or in the gamete. So in this case, since we are talking about diploid, so number of autosomal sets in the gametes is 1, right? So it is minus 1. So number of X chromosomes here, it is 3. So 3 minus 1, 2. So 2 will be the number of bar bodies in this case. So if they are giving it for diploid, what if they are asking for polyploid? For example, 2n is equal to 4x, which is equal to any number, 40 let us take. So what will be the autosomal sets in gamete? It will be either 1 or 2 if it is 4x. So it will be 2, right? So in that case, the answer would be 3 minus 2. Only one bar body will be formed if it is the case of tetraploid. If they are asking for tetraploid or polyploid, you have to apply the formula where it is number of X chromosome minus number of autosomal sets in the gametes. Or if they are directly asking for diploids, you not to do anything, it is just simply number of X chromosome minus 1. It is always minus 1 in case of diploids. For polyploids, it depends on the ploidy. So the general formula will be number of X chromosomes minus number of autosomal sets in the gamete. That's it. The last one. So in the population, if a population was subjected to selection in the favor of heterozygotes for 10 generation, so how many generation of random mating will be required to restore the equilibrium for 2 alleles or a single gene? So I have already discussed this concept in the earlier one. So I will be expecting the answer from your end. So comment your answers on this video. So if you do any mistake, I will definitely correct it. Okay. So you have to take it in a sporty way and you have to answer this question in the comment. So I hope like you have understood previously what I have explained regarding the concept of restoring the equilibrium, heterozygotes, and about the generations and the genes. So this is the part 2 of genetics. So we'll be also coming up with part 3 for genetics. So we have left with some more questions. We will do it in the part 3, and then we have part 4 for plant breeding. Okay. So it's my kind request to you all, if you like the content, if it is helping you, like, comment and share this video. So we'll be coming up with the other videos as well on a regular basis. So if you just encourage us, we'll be definitely doing it on a daily basis. Okay, thank you.